This is a small point that I missed. It's just that it's kind of self-explanatory, but yeah, enzyme lowers activation energy. I mean, that's kind of self-explanatory. With enzyme, you see this much of activation energy is reduced. You're only needing this much of activation energy. So yeah, okay. Classification and nomenclature of enzymes. Thousands of enzymes have been discovered, isolated, and studied. Enzymes are divided into 6 classes, each with 4 to 13 subclasses named accordingly by 4-digit number. Just know this. Oxidoreductases or dehydrogenases. Enzymes which catalyze oxidoreduction between two substrates. So basically, this substrate is reducing and this substrate is oxidizing. Oxidoreductases or dehydrogenases just catalyze, make the reaction fast, that this S oxidizes and this reduces. That's the whole point. Transferases, enzymes catalyzing a transfer of a group. So in this between, okay, here you have Sg plus S dash. This in the presence of transferases fastens, and S plus S dash G. This G group is going to the S dash, which was formerly on the S. Hydrolysis, enzymes catalyzing hydrolysis of ester, ether, peptide, glycosidic C halides, P and bond. Okay, so hydrolysis, basically I told, right, glucose. Okay, let's say sucrose, glucose plus fructose. When you're hydrolyzing, glucose and fructose are connected by glycosidic bond. When you're hydrolyzing, when you're performing hydrolysis in the when you're hydrolyzing, that in the presence of hydrolysis, which is fastening the reaction, that the bond breaks, so you get glucose and fructose. That's the whole point. So these are catalyzing hydrolysis. The point is lysis or whatever. Enzymes that catalyze removal of groups from substrates by mechanisms other than hydrolysis, leaving double bonds. Okay, so you have CX, CY. Now when you want to remove this, you want X plus Y, you will have to suffice the valency of C. Here let's say you have H. Okay, now you have only 2H here, like so you need a double bond, so you're getting a double bond, and you're removing this. It's not hydrolysis. That's what is the point here. Isomerases is a very important one. Okay, so you have, it includes all enzymes catalyzing interconversion of optical, geometric, or positional isomers. I'll give an example. You have, okay, C double bond C, COOH, H, COOH, H. This is one. This is another. Okay, this one you just leave for now. Okay, these compounds are structurally, okay, let's just see, they are having the same, like, you know, molecular formula, CCOH, COH, 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 H, C double bond, C double. The only thing that's changing is the position of COH. It's like the orientation of COH in the plane, or wherever it's orienting in the, you know, that is changing. When you're converting this into this or this into this, you're using some isomerase. Okay, this isomerase includes all enzymes catalyzing interconversion of optical, geometric, or positional. These are geometric isomers. Okay, this is a cis one second, cis-butene dioic acid. I'm sorry, okay, this is cis, sorry, this is trans cis. Okay, the cis one is called malic, like, okay, you just see here, malic acid, and this is fumaric acid. Okay, these are geometric isomers. So the interconversion between them, between isomerases. Ligases, this we will learn in biotechnology. We have DNA ligase. Okay, this DNA ligase connects two fragments of DNA. So ligases are basically linking together two compounds. Enzymes catalyzing the linking together of two compounds. Example, enzymes which catalyze joining CO bond, CS bond, CN, PO, etc. bonds. Cofactors, enzymes are composed of one or several polypeptide chains. Okay, you know, enzymes are proteins. Okay, enzymes are proteins, like they have polypeptide chains, but there are also some polypeptide chains that are not proteins. Okay, they are not forming any proteins. They are called non-protein structures, or non-protein, whatever, non-proteins. They they are the non-protein constituents. And when some enzymes, they have proteins as well as non-proteins. Like, okay, this is not enzyme. Basically, enzyme has a protein part and a non-protein part. The protein part is called apoenzyme, and the non-protein part is called cofactor. I'll explain here. Okay, so here there are number of non-protein constituents called cofactors are bound to the enzyme to make the enzyme catalytically active. Okay, so in these, the protein portion of the enzymes is called the apoenzyme. Okay, 3 kinds of cofactors may be identified: prosthetic group, coenzymes, and metal ions. Okay, the prosthetic, okay, you understood here, like basically your cofactors are required for the catalytically active nature of enzyme. Without them, without the cofactors, the enzyme cannot catalyze. Okay, and how did we know this? Of course, when we removed the non-protein part, the cofactor part from the enzyme, and saw whether it was catalyzing, we saw that it did not catalyze. So then we understood that non-protein parts, the cofactors, play a crucial role in catalytic activity of enzyme. That's the last part. Prosthetic groups are organic compounds and are distinguished from other cofactors in that they are tightly bound to the apoenzyme. So your prosthetic groups of the cofactors. Cofactors are divided into two, organic and inorganic. In organic, you have two, prosthetic and coenzymes. In inorganic, you have only your metal ions. So cofactors, the prosthetic, they're tightly bound. So these are always bound. Okay. So for example, in peroxidase and catalase, which catalyze the breakdown of hydrogen peroxide to water and oxygen. H2O2 peroxidase, I didn't write the full, H2 plus O2. Heme is the prosthetic group. This is the peroxidase. Okay, whatever. That is the enzyme. This is the heme, is the your cofactor, your prosthetic group, and it's a part of the active site of the enzyme. Okay. Coenzymes are also organic compounds, but their association with the apoenzyme is only transient, usually occurring during the course of catalysis. So coenzymes serve as cofactors in a number of catalyzed reactions. Okay. So what coenzymes do is that only when catalytic activity is happening, the enzyme is catalyzing something, they are there. The coenzymes are there only when catalysis is happening. In other times, they are free. Okay. So for example, coenzyme nicotinamide adenine dinucleotide (NAD) and NADP contains vitamin niacin. Usually, the essential chemical components of many coenzymes are vitamins. So NAD and NADP have niacin vitamin. That's what they're saying. And the number of the metal ions, what they do is that they form coordination bonds with side chains as well as with the substrate. Okay, they form one or more coordination bonds. Example, zinc is a cofactor for the proteolytic enzyme carboxypeptidase. This is something in relation with protein. Okay, of course, proteolytic enzyme carboxypeptidase. Catalytic activity is lost when the cofactor is removed from the enzyme, which testifies that they play a crucial role in the catalytic activity of the enzyme. And that's the whole. Let us see if there's anything in summary. Okay, if you want to see this we know, of course. 20 types of amino acids. Enzymes lower activation energy of reactions and enhance greatly the rate of reaction. I think that's the whole thing. Okay, so yeah.